everybody. Welcome to our final session of the 42nd National Nutrient Data Bank Conference. We have had 3 days of wonderful presentations, and this last one as well will not disappoint. I'm Alanna Moshfegh. I'm with the Beltsville Human Nutrition Research Center at the Department of Agriculture. Session 9 this afternoon is on dietary assessment of glycome. Our first presenter will be Nikita Bacalzo, who's a fourth-year PhD student at the University of California in Davis in the Department of Chemistry. He will be presenting on Glycopedia, a Glycan encyclopedia of food. Our next two presenters are with the Agriculture Research Service Western Human Nutrition Research Center, also located at the University of California in Davis. Our first presenter will be Jules, our second presenter will be Jules Larke, who's a computational biologist at the Western Human Nutrition Research Center. His presentation will be on sources of specific monosaccharides from foods and ingredients commonly consumed by healthy adults. Our final presentation for session 9 will be from Danielle Lemay, who's a research scientist at USDA's Western Human Nutrition Research Center. Her presentation will be on surveying nutrient assessment with photographs of meals, or SNAPMe, a data set of food records and companion meal photos. So let's begin.
Hello, everyone. First of all, I would like to thank the organizers of this conference for allowing me to share our project with everyone. Our team has recently created a Glycan-focused database of foods, and I am excited to share more about this project to you all. I am Nikita Bacalzo. I'm a graduate student from the Library Lab from UC Davis, and I will be talking about the Davis Food Glycopedia, a Glycan encyclopedia of food. It is common knowledge and has been proven many times over the centuries that food and human health are highly intertwined. Starting from the gestational period during pregnancy, during our infancy, and with adult life, our diet has a major impact to our overall health. However, to this day, there are still gaps in our knowledge about the components in our food. And as an analytical chemist, we aim to fill these gaps, and we try to develop new methodologies to gain deeper understanding of the food that we eat. And one of the shortcomings in food analysis revolves around food carbohydrates. Our lab and our collaborators has shown that oligosaccharides present in human breast milk serves multiple functions for the infant through the gut microbes. These oligosaccharides are not digested and absorbed by the infant, but rather it accumulates in the lower gastrointestinal tract to serve as decoys for some pathogenic bacteria and also to serve as fermentation substrates for health-associated commensal bacteria. As we grow older, our diet changes as well. We start to eat more diverse foods such as the plant-derived ones. During this transition, our gut microbes also adapt to these changes. In the recent years, it has been shown that food carbohydrates, specifically polysaccharides, modulate the gut microbial community, not only in terms of taxonomic abundance, but also in fermentative function. These microbes are generally equipped with enzymes capable of degrading and utilizing various polysaccharides as their main carbon source. And so the goal of these gut microbiome focused studies is to understand how food can affect human health through gut microbiota interactions. And for these to be feasible, detailed characterization of food structures is necessary, so we may be able to correlate them to the gut microbiome and then eventually to its downstream effects on the human health. Plant polysaccharides are a major source of dietary fibers. Different plants and plant parts produce these diverse polysaccharides, which mainly serves as energy storage and also to provide structural integrity to the plant. Carbohydrates, specifically dietary fiber, are based on bulk gravimetric data. An example of a workflow for dietary fiber measurement is shown here. The first step usually involves enzymatic digest using proteases and amylases to digest proteins and starch, the remainder of which will be labeled as total dietary fiber. These can further be separated into insoluble and soluble dietary fiber fractions using alcohol precipitation. While this method is validated, it is rather low throughput and specific structures are not determined. Different enzymes can be used to analyze different polysaccharides. However, scaling this to larger sample size and also to multiple polysaccharides would be impractical. To address this lack of specific structures from current carbohydrate workflows, our lab has been developing new methodologies to gain more structural and quantitative information on food carbohydrates. One of these is the rapid throughput of monosaccharide analysis. So the workflow starts with homogenizing the food sample in an aqueous solution and then transferring them into a 96-well plate, from which we add a cocktail of enzymes to enhance our coverage of some of the pectin polysaccharides, and then it will undergo hard acid hydrolysis to fully digest all the polysaccharides into their constituent monosaccharides. And then lastly, we chemically label it with a chemical tag called PMP, and this serves as two functions: one, to enhance the separation in the liquid chromatography, and two, enhance the signal from the mass spec. The analytical platform that we used is a UPLC with a C18 column coupled to a triple quad mass spectrometer. Using this platform, we are able to monitor the 14 most abundant monosaccharides in nature, each of which are well resolved in both the LC and M/Z dimensions. So we have applied the rapid throughput monosaccharide analysis to one of our food biomarker projects, the overall goal of which is to find biomarkers associated with specific food microbe interactions. And specifically, these compose of three major aims, the first one of which is to develop the carbohydrate encyclopedia for feeding studies, which will inform the other two sub-projects, namely to use fecal bioreactors and to do a clinical trial feeding study on a cohort of toddlers. So for the selection of foods to be included in the Glycopedia, first, we selected fiber-rich single foods that were recommended for toddlers aged 12 to 36 months according to the 2020-2025 Dietary Guideline for Americans. And then next, we also purchased commercially available complementary toddler foods as well as infant foods. And then lastly, we populated the Glycopedia with commonly consumed foods by adults in America using three different databases. These food samples were purchased locally from grocery markets as well as from online suppliers. And at the end, a total of 822 foods were analyzed for their monosaccharide profile. Additionally, we have manually curated the metadata associated with this database, and this is just a list of all the information that we have regarding for each sample. So we have information like the product storage upon purchasing, and also the packaging form, variety or cultivar of the product, and so on. So this graph shows the distribution of the Glycopedia by food groups. And as we can see, a lot of the samples were fruits and vegetables. And then there were also a lot of green products and then beans, peas, other legumes, nuts, and seed. The other categories of foods were significantly lower than the four groups that I've mentioned. These plots are example chromatograms of actual food samples and their monosaccharide profile. And for reference, we also have the pooled monosaccharide standard here. So as we can see, we are able to detect highly abundant monosaccharides as well as the low abundant one. These peak area abundances are converted to absolute quantity using external calibration curve. After analyzing the 828 food samples for their monosaccharides, we aggregated their values according to their food groups, and this is summarized in these graphs. So the most commonly found and often the most abundant monosaccharide was glucose, which was likely from starch or sucrose, which unsurprisingly is present in a lot of our food samples. And the other common and abundant monosaccharides that we detected were fructose, xylose, Arabinose, galactose, and some galacturonic acids. Xylose, Arabinose, galactose, and galacturonic acids are likely from cell wall polysaccharides such as arabinoxylans and pectins, and specifically for grain products, xylose and arabinose are most likely from arabinoxylans, while in fruits and beans, peas, legumes, nuts, seeds, and some vegetables, galacturonic acid and rhamnose are likely due to the abundance of pectins from these samples. Overall, the grain products group had the highest measured carbohydrates by fresh weight due to their high starch and also low moisture content. The groups of eggs and also the groups with fats, oils, and salad dressings were found to have the lowest carbohydrate content, while the peat poultry fish mixture group contained significant amounts of glucose, largely due to the bread coatings of some of these samples. However, the analysis of unprocessed meat, poultry, and fish revealed very little glucose. We can then zoom in into this food groups and see some of the variations within each food group. For example, shown here are some examples of fruits in panel A, where we see that glucose and fructose are the dominant monosaccharides, and these are mostly coming from sucrose and their free monosaccharide forms. And for banana, we expect them to have a higher starch than the other fruit samples. Other non-glucose monosaccharide include xylose, arabinose, and galacturonic acid, again as expected, as these fruits contain pectins. For the grain products, these all have relatively higher amounts of glucose coming from starch, and again, some have relatively high amounts of xylose and arabinose coming from the arabinoxylan. And then looking at examples from the vegetable food group, we see more diverse profiles. So aside from glucose, we can also see fructose, galactose, xylose, arabinose, some galacturonic acid, and for a few samples, we also see a relatively higher amounts of mannose. And then lastly, for this group here, which are the beans, peas, other legumes, nuts, and seed group, we actually see relatively higher amounts of arabinose compared to the other food groups. And then we also observed that specifically beans and peas had larger amounts of glucose due to their higher starch content. We did unsupervised hierarchical clustering based on the monosaccharide composition of the 828 foods. The number of clusters shown here was chosen based on some clustering optimization metrics that were to maximize inter-cluster variance while also minimizing intra-cluster variance. So the blue heat map here shows the log transform monosaccharide value where each track corresponds to each monosaccharide as labeled here. The outermost track corresponds to the food group based on the color legend here. And then the innermost track is the dendrogram where 5 clusters are shown. We have observed that to some extent, food samples clustered based on their food groups. For example, clusters 2, 4, and 3 on this side of the heat map. If we look at the outermost track, we see that these are mostly brown colored samples, and that corresponds to the grain products. We further analyzed these clusters based on enrichment of some of the food groups, and we have found that statistically, cluster 1 was enriched in fruits, veggies, and the beans food groups, while clusters 2, 3, and 4 was statistically enriched with grain products. And then looking at the aggregate monosaccharide profiles of each cluster, we noticed that the clustering was mainly driven by the glucose abundance. As we can see here, the glucose increases from cluster 1 to cluster 4. And then cluster 5 actually is a single member cluster comprised of coconut flour, which has the highest amount of mannose in the Glycopedia. Cluster 2 contain mostly breads and cooked whole grains such as oats, barley, millet, quinoa, and rice. And then cluster 3 are the more dried products like cereals and some dried snacks. And then cluster 4 contains 6 items, all of which were either dried rice or dried corn products, exhibiting the highest glucose and the lowest non-glucose monosaccharides of all of the foods analyzed. The idea of personalized nutrition is not new, but creating food combinations based on monosaccharide data is novel. For example, you can have a certain meal, and using the Glycopedia data, we can now assess how much of each monosaccharide are you eating with your food. And this level of resolution in food data is exactly what is needed in clinical feeding trials in the gut microbiome research. When we look at Arabinose contents of food, we observe that Arabinose is relatively high in beans, peas, legumes, nuts, seeds, as well as also in grain products. It is found commonly only in plants. It is also not digested or absorbed well endogenously in animal models, and it has been shown to play an important role in shaping the gut microbiome. Arabinose is not abundant in foods as a free monomer. Rather, it is part of a ubiquitous cell polysaccharides such as arabinoxylan, which are highly abundant in grains, and pectins, which are found in fruits, veggies, beans, and other legumes. To summarize, we have created a Glycan-focused database which was enabled by a high-throughput monosaccharide analysis workflow. We envision that these Glycopedia will help improve and guide study design in clinical feeding trials, especially in the gut microbiome-focused studies. Right now, we're currently further expanding the coverage of the Glycopedia data and apply our other glycomics workflow to include glycosidic linkage composition as well as polysaccharide composition in the near future. And with that, I would like to thank and acknowledge all the people who have been involved in this big project. I would like to thank Professor Carlito Rubia for allowing me to work on this project, as well as the members of our lab who have contributed greatly to this project. Juan, Dave, Winnie, Garrett, Ryan, Charlie, Aaron, Chris, and Sean, and also our collaborators from the NIH Food Biomarker Project, Dr. Mills, Dr. Bruce Sherman, Dr. Smilowitz, Dr. Lemay, Dr. Chin, Dr. Larke, Dr. Kalnetra, Dr. Sitapu, Sarah, Yasmin, Yael, and Shad, and also our undergraduate students who help us in generating the, the sample set and also during the data analysis. And lastly, we would like to thank our funding sources, specifically NIH for this project, and also USDA, Mars Mondelez International for funding some of our projects in our lab, and then also the UC Davis Department of Chemistry. With that, I will be happy to entertain some questions. Thank you.
Hello, and welcome to this portion of the dietary assessment of glycome in the National Nutrient Databank Conference. My name is Jules Larke, and I'll be talking about sources of specific monosaccharides from foods and ingredients commonly consumed by healthy adults. To provide some background, carbohydrates are a major component of dietary intake, providing a significant source of calories from starches and sugars, and in the case of non-digestible carbohydrates, carbon sources for our gut microbes. However, current assessment of carbohydrate intake is limited to estimating simple sugars, starch, and dietary fiber, based on the more commonly available databases. This is a limitation as these broad categories lack the specificity to capture the molecular detail and diversity of our food. Recent advances in analytical chemistry have made it possible to analyze the monosaccharide composition of foods in high throughput. And so by leveraging fine-scale food composition data, we can more accurately describe the relationship between diet and health outcomes, as well as devise new predictors of dietary assessment. In this presentation, I'll be going through my main study objectives, first of which is to ingredientize dietary recalls for mapping to the food Glycan database. Next, to characterize the monosaccharide composition of diets in a healthy US cohort. And then lastly, to investigate the relationship between monosaccharide intake and diet quality. The participants for this study were taken from the USDA Nutritional Phenotyping Study. To briefly describe the cohort, participants were recruited in an 18-bin sampling scheme balanced for age, sex, and BMI. As this study was designed to investigate healthy subjects, individuals were excluded if having recently undergone a major surgery or receiving antibiotic therapy. Additional exclusion criteria includes those taking medications for a diagnosed chronic illness, including diabetes, cardiovascular disease, GI disorders, hypertension, and several others. And now to describe the Food Glycan database, also known as the Food Glycopedia. This database consists of 913 foods across 9 food groups. Here you can see a distribution of the foods in these food groups. For each of these foods, the Glycopedia has absolute values of 14 monosaccharides obtained using HPLC-MS. Here you can see a list of the 14 monosaccharides. For the dietary assessment portion of this study, we used automated self-administered 24-hour recalls, or ASA-24 system, to assess recent dietary intake. Recalls were then merged with the Food and Nutrition Database for Dietary Studies, or FNDDS, version 2017-2018, to retrieve ingredient level information. And I'll give more information on this in the following slides. Participants with less than 75% of their carbohydrate intake mappable to the Glycopedia were excluded in the final analysis. And so moving on to our first study objective, we're going to discuss ingredientizing dietary recalls for mapping to the food Glycopedia. The main challenge of trying to achieve high coverage of diet recalls mapped to the Glycopedia is that there are more unique foods consumed by the cohort compared to the number of foods in the Glycopedia. So, I took the approach of ingredientizing mixed dishes into their components to reduce the number of unique food descriptions. Doing so increased the coverage of diet recalls that could be mapped to the Glycopedia. For example, there are several mixed dishes that are a combination or subset of a list of ingredients. Take, for instance, pizza, which contains tomato sauce, wheat flour for the dough, mozzarella cheese, and pepperoni. We can use this ingredients list to derive other mixed dishes such as pasta, as it also contains wheat flour, tomato sauce, and cheese as main ingredients. Additionally, as the Glycopedia mainly consists of single ingredient foods rather than mixed dishes, this approach increases the overall mapping to ingredientized diet recalls. Here I'm describing how the process of ingredientizing diet recalls affects the total number of foods and the total number of unique foods in the dataset. So if we start on the left side of this, we can see that the QC'd recall data consists of 341 participants over around 1,000 recalls. These recalls have 19,000 total items that spanned 2,200 unique food codes. After extracting the ingredients with FNDDS, we see that the ingredientized dataset has expanded the total number of items to 45,000. However, this has reduced the total number of unique ingredients or unique foods down to 1,199. Following this, filtering participants based on a criteria of having less than 75% of their calories from carbohydrate mapped to the Glycopedia, our final dataset consists of 180 participants over 500 recalls, with 14,000 total items and 465 unique ingredient codes. Those ingredient codes are then mapped to the Glycopedia to 241 distinct foods. Here we're looking at a correlation between the total monosaccharide and total carbohydrate consumed in the cohort, with each point representing one subject's average intake across recalls. This acts as a reality check of our assumption that the monosaccharide and carbohydrate intake are similar despite being derived differently. So for example, on the y-axis representing the total carbohydrate intake, this value is derived from FNDDS with the standard reference value. And this is a by difference type of metric, meaning that it's taking into account the difference between 100 and the sum of the percentages of the remaining measurable components in the food. Contrast that with the x-axis, monosaccharide values, which are analytically derived by HPLC-MS for each food item. Moving on to our study objective number 2, we're going to characterize the monosaccharide compositions of diets in this healthy U.S. adult cohort. Here I'm showing the glycan composition of the diet in the cohort by food category. In this stacked bar chart, we can see the average monosaccharides consumed across recalls on the x-axis. The most noticeable thing here is that glucose comprises a majority of each food category. If we take away glucose and just look at the average non-glucose monosaccharides consumed across the cohort, we can see some more interesting trends. For example, vegetables and fruits at the top here have a higher amount of GAL-A. GAL-A is a component of pectin fibers, and these pectins are highly abundant in vegetables and fruits. In addition, if we look at grain products, grains have a higher amount of Arabinose and xylose, reflecting the arabinoxylane content in the dietary fibers of grains. Here we're looking at the top monosaccharide contributors by individual food or ingredient. The x-axis for each of these plots represents the total amount of each monosaccharide consumed across the cohort and scaled to 1,000 kcals per participant. If we look for the top contributors for glucose, we can see that is enriched long grain white rice, yellow banana, and then flour tortillas. For galactose consumed, this is all represented by dairy products, which makes sense as it reflects the lactose content in these foods. fructose consumed, we can see that apple and then banana and a soft drink make up the top contributors. Breaking down the monosaccharide intake across the cohort, we can see that on average, glucose made up a majority of the intake, unsurprisingly. This is around 80%. These are then followed by fructose and galactose, each coming in at around 5 to 6%. This is followed then by arabinose, xylose, gal-A, and mannose. The remaining 7 monosaccharides that were measured make up less than 0.5% of the cohort's diet on average. Moving on to the final study objective, we're going to investigate the relationship between monosaccharide intake and diet quality. If we examine the monosaccharide intakes by participants, we see a broad range in glucose consumption. As we know, glucose is a component of some dietary fibers. However, given the grams of glucose intake, those on the higher end are likely consuming a majority of glucose in the form of starch or simple sugars. Conversely, many of the non-glucose monosaccharides are more typically incorporated into dietary fiber. And so, with this observation, it seems relevant to investigate the relationship between diet diversity of monosaccharide intake and diet quality. Using Shannon diversity of monosaccharides as a proxy for diet diversity, we've compared this with the healthy eating index scores taken from the participants. Briefly, the HEI score reflects how well a diet aligns with the dietary guidelines for Americans. Here we can see that there is a positive correlation suggesting a diet more varied in monosaccharide content corresponds to a healthier eating pattern. Overall, concluding that higher monosaccharide diversity reflects a varied source of fiber intake from increased consumption of non-digestible carbohydrates. In summary, I'd like to mention a few takeaways. This is the first study to investigate population-based dietary intake at this resolution of food glycan composition. Fine-scale food composition is needed to better characterize dietary intake so that we may tailor personalized dietary recommendations. And then lastly, as we've seen, greater diversity of monosaccharides in the diet corresponds to a healthier eating pattern, which may be attributed to reduced intake of simple sugars. In concluding, I'd like to make a few acknowledgements. First, from the USDA-ARS at WHNRC, Elizabeth Chin, Charles Stevenson, Danielle Lemay, as well as the entire nutritional phenotyping study team. Without their help, this would not have been possible. Secondly, at UC Davis, the Labria Lab has been instrumental in gathering so much data tirelessly for construction of the Glycopedia. So thank you. Lastly, the funding sources from the USDA, NIH, as well as the Artificial Intelligence Institute for Next Generation Food Systems. Thank you all so much, and I look forward to your questions.
Hi, I'd like to thank the organizers for inviting me to speak with you today about our study entitled Surveying Nutrient Assessment with Photographs of Meals, a data set of food records and companion meal photos. First, I'd like to step back and help you understand the motivation for this. Our overall goal is to estimate dietary glycans and other food components in the wild, meaning in free-living humans. And to do that, we need to capture foods and ingredients. I probably don't need to explain to this audience why our dietary assessment tools need modernization, because as we all know, they have high subject burden, they're very error-prone, they're not particularly quantitative, and they don't have the resolution that we need for research. In particular, much of nutrition is what we think of as dark matter. So today, when we get data from a food frequency questionnaire or a 24-hour dietary recall, there's sort of a limited number of nutrients that we get as output. And by limited, I mean there's still 70. It's still quite a lot. But there are thousands of molecules in food, some of which we just don't know yet. And we'd like to be able to capture the actual foods and ingredients that are being eaten so that we can, as more data comes online, we can map to those foods and ingredients. And you learned today about one such special interest database, the Davis Food Glycopedia. It's certainly not the only special interest database, but it's one that's very important for understanding diet microbiome relationships. And in order to map foods that are being consumed by participants in research studies to this database, we need to capture foods and ingredients. So that brings me to our next topic about needing a benchmark. One way of modernizing our dietary assessment methods is to move towards photo-based diaries. And it's pretty easy for participants to take a picture. Many of them who are not even in studies are taking pictures of their food all the time. But we have to ask ourselves, how good are these mobile food records? And as far as energy intake is concerned, there have been studies to look at that, and there was recently a systematic evaluation of mobile food records. Energy and macronutrients have been surveyed. In general, they're underreported by mobile food records, but these have been tested in kind of smaller populations. We need tests in larger populations, specifically looking at more than just the energy and macronutrients. So we really need to evaluate food prediction, not just nutrients. And we need to be able to predict those foods so that we can map to special interest database. So for example, taking a picture, an algorithm converts that picture into ingredients, and then those ingredients can then be mapped to any database. It could be USDA's Food Data Central or any database that has not yet been incorporated into Food Data Central. So the field really needs a benchmark. And so that's basically a common standard against which all food and ingredient prediction algorithms can be measured. This benchmark needs to contain both food photos and ground truth of what is in those photos, and ideally collected in the context of dietary assessment. So now I'll tell you about the SNAPMe benchmark. So SNAPMe stands for Surveying Nutrient Assessment with Photographs of Meals. The goal was to develop a reference data set of 1,000 real-world meal images captured in parallel with a traditional method, so that this benchmark could then be used to evaluate computer vision algorithms. So we recruited participants to use both the BiteSnap food photo diary and in parallel to record their diets using the ASA24 in food record mode. We recruited participants from among those who had, previously participated in research, and we recruited, athletes. We recruited through the undergraduate nutrition and food science programs throughout the country, the top programs, as well as individuals we could reach out to through social media or cultural groups to increase the diversity of the dietary records. Overall, though, we were really concentrating on trying to enrich our participant pool with what I'd like to call food nerds, because we're we're creating a ground truth database. So the screening process, basically participants had to be adults who had a smartphone, were not pregnant, and were willing to record what they're eating for 3 days, and without histories of eating disorders or wide swings in weight loss, and willing to eat at least 3 meals a day that were varied. And perhaps the most important part of our screening process is we wanted to make sure that people were able to accurately record what they were eating, and so we put them through tests as an enrollment criteria, and showed them pictures of various meals and asked them to answer which ingredients they saw in those meals, and we developed metrics for what we considered was a pass. And likewise, we also asked people to estimate portion sizes, and so people had to do well enough on these tests in order to be enrolled into the study. Then once those who qualified were enrolled, we had an enrollment meeting in which they were taught how to use the BiteSnap app and how to log those foods with the ASA24. Then for the data collection phase, as they were collecting photos, they would take different types of photos. Either it was not packaged food, food that they had prepared at home, or maybe eaten at a restaurant. Those would be displayed along with a fiduciary marker, this size selection marker that you see here, the black and white checkered marker. Or if they had a packaged food, then they were instructed to take a photo of the front of the package as well as the ingredients label of that package. And each of these types of images then were paired with particular food items within the BiteSnap app and the ASA24. So the data was initially put through quality control in semi-real time, like as participants were uploading their data each day, someone would that day review their ASA24 just to make sure it's complete and within a reasonable range of calories and at least 3 meals. And they would review their BiteSnap photos to make sure the photos were clear and appropriately labels viewable, and there's no personal information, and the sizing marker is included. And if for some reason a participant did not do well at this, they were actually asked to redo the day, a different day, because our objective wasn't, accurately assess the diet of that particular individual. It's to produce a ground truth data set. So in total, 96 participants completed the trial. We have 3 days of data from 90 of those participants. Overall, there are 279 days of dietary data that have been collected and more than 1,000 images collected. So then after the all enrollment was complete, people had completed the trial, then the longer process of QC and database preparation began. And this was a manual review of all ASA24 records and BiteSnap photos. And we prepared linkage files to link each ASA24 food entry to each BiteSnap photo. So both the ingredient and quantity and the ASA24 to which BiteSnap photo. And these, the images together with these linkage files make up the benchmark. Okay, so what is next? So our next steps is we plan to evaluate the performance of a computer vision algorithm, probably Facebook inverse cooking, on the food and ingredient prediction using this SNAPMe benchmark. And we plan to release the SNAPMe benchmark and manuscript to the public, aiming for this fall. And beyond that, of course, hoping to improve computer vision algorithms. So for the future, we the SNAPMe project, the SNAPMe benchmark, we plan to be useful in at least two ways. One is to be able to evaluate existing algorithms and apps that are on the market for an apples-to-apples comparison. And the second is to evaluate new computer vision algorithms for improvements in predicting food and ingredients. So there are many, many people involved in this project, but I'd like to highlight a few very important people. So Dr. Elizabeth Chin was the technical lead on the computer vision aspects of our overall research program, and she really pushed starting this study and carrying through its completion. And Dr. Jennifer Smilowitz was the clinical study PI of SNAPMe, and we had two very hardworking study coordinators, Yael Vainberg and Yasmin Vasib. And certainly lots of help from BiteSnap's Mike Walski. And that concludes this talk, and I look forward to your questions in the panel. Thank you.